Hello students, today we study a new chapter, Human Health and Diseases. According to World Health Organization, health is defined as a state of complete physical, mental and social well-being. Though we are exposed to different foreign bodies like bacteria, viruses and a variety of pathogens, most of us remain healthy, and this is due to human body has ability to resist almost all types of these foreign bodies. Related with health, we study some important terms, that is about the immunity and immune system of body. So first study the immune system. The system which protects us from various infectious agents is called immune system. Then C, immunity. Actually, immunity is nothing but resistance power of our body. So, immunity is resistance exhibited by the host towards injury caused by pathogens and their products. Then, the term immunology. It is a branch of biology or branch of science which deals with the study of immune system and immune responses to foreign substances and their role in resisting infection by pathogens. Actually, there are some important characteristics of immune system, that is, it distinguishes self, that means within the body cells or body's own cells, and non-self, that is foreign molecules or invading cells. म्हणजे बाहेरून आलेले काही घटक, म्हणजेच त्याला नॉन सेल्फ म्हटलेलं आहे. Any foreign substance invading in body and capable of stimulating an immune response is called antigen. And the protective chemicals produced by immune cells in response to antigens are called antibodies. म्हणजे अँटीजेन आणि अँटीबॉडी म्हणजे काय हे अगोदर आपल्याला समजून घेणं गरजेचं आहे आणि त्याच्या डेफिनेशन्स लक्षात ठेवणं पण गरजेचं आहे. देन वी स्टडी the types of immunity. So, see in the diagram, simple. Immunity is of two types: innate immunity, acquired immunity. Then we study actual types of immunity. Immunity is two types, main two types. First one is the innate immunity. It is also known as inborn immunity, or it is also known as inherited immunity. So see in the diagram, a type of innate immunity which is always on, which gives immediate response, which contains complement system. It also contains antigen presenting cells and number of different types of barriers. So, simple few characters of innate immunity. It is natural defense system of body. Innate immunity is the resistance to infections that an individual possesses virtue of his or her genetic makeup. Therefore, it is inherited immunity. It is not affected by prior contact with microorganisms or immunization. It is non-specific when it indicates a degree of resistance to infection in general, or it is specific where resistance to a particular pathogen is concerned. Innate immunity comprises different types of barriers, and one form is we study, and that barriers which prevents entry of foreign agents into the body. So, see the barriers. First barrier, that is epithelial surface. And in epithelial surface there are number of components such as intact skin and mucous covering of the body protects against invasion by microorganisms. Healthy skin possesses bacterial activity due to the presence of high concentrations of salts in drying sweat. Then sebaceous secretions and long chain fatty acids have bactericidal and fungicidal properties. Mucosa of respiratory tract has several innate mechanisms of defense. Particles that are entered through the inhaled air being arrested through the hairs in the nasal orifice. एक्सटर्नल नॉस्ट्रिल्समधनं काही foreign particles inhaled होतात. These foreign particles that pass the nasal orifice and held by mucous lining, these are also swept back to the pharynx where they are swallowed or coughed out. दोन्ही प्रोसेसेस इथं होऊ शकतात. Thus, the cough reflex is important defense mechanism of respiratory tract. The mouth is continuously and constantly bathed in saliva, which has inhibitory effect on microorganisms. Then the acidity of gastric secretions in stomach destroys microorganisms. Flushing action of urine eliminates bacteria from the urethra, and spermine and zinc present in semen or seminal fluid are acts as these are antibacterial. In this way, there are number of components in the epithelial surface. Then second barrier is antibacterial substances in blood and tissues. Complement system contains more than 30 serum proteins which are circulating in blood, but these are in inactive state. Microbial pathogens activates the complement cascade to eliminate pathogens. And interferons, which are a class of cytokines. Cytokines, for example, soluble proteins released by cells infected with viruses, and the WBC stimulates other cells to protect themselves from viral infection. The next barrier is cellular factors in innate immunity. Foreign particles is mediated by phagocytic cell. Generally, these cells are grouped as microphages and macrophages and remove foreign particles from body. Natural defense of the body against invasion of blood and tissues from microorganisms and foreign particles is mediated by phagocytic cells, which ingest and destroy the foreign microorganisms. Lymphocytes, commonly known as natural killer cells, which are important in non-specific defense against viral infection and tumors. Next barrier is fever. Increase in the body temperature following the infection is a natural defense mechanism. It helps to accelerate the physiological processes to destroy invading pathogens, and fever stimulates the production of interferons and helps in recovery from viral infection. Next barrier is acute phase proteins, or these are simply known as APPs. Infection on injury leads to a sudden increase in certain plasma proteins, and these are collectively called as acute phase proteins. These proteins include, for example, C-reactive protein, commonly known as CRP, mannose binding protein, alpha-1 acid glycoprotein, and serum amyloid, etc. Thus, APPs are believed to enhance host resistance and prevent tissue injury and repair of the inflammatory lesions. Then actually we study the second type of immunity, that is the acquired immunity. See in the diagram, immunity, second type of immunity is acquired. It again divided into two types, active and passive. In active there are own antibodies, in passive there are ready-made antibodies. These immunity systems again divided into natural and artificial. But before going in detail the types, first we study the some important characteristics or unique features of acquired immunity. This immunity involves formation of antibodies in the body which neutralizes antigens. जे काही इन्फेक्शन आपल्या शरीरात झालं असेल इन द फॉर्म ऑफ अँटीजेन्स ते न्यूट्रलाइज करण्याचं काम ॲक्वायर्ड इम्युनिटी करते. आणि त्याच्यासाठी त्या सिस्टीमचे ॲक्वायर्ड इम्युनिटीचे काही युनिक फीचर्स आहेत. हे युनिक फीचर्स फार महत्त्वाचे आहेत. फॉर एक्झाम्पल, फर्स्ट feature, that is character, specificity. It can produce specific antibody or T-lymphocytes against a particular antigen or pathogen. ह्यालाच specificity म्हणतोय, विशिष्ट प्रकारचं. Then second one is the diversity. It can recognize a vast variety of diverse pathogens or foreign molecules. Then next one is discrimination between self and non-self. सेल्फ आणि नॉन सेल्फ मगाशी आपण बघितले की सेल्फ म्हणजे स्वतः आपल्या बॉडीमधल्या आणि नॉन सेल्फ म्हणजे फॉरेन मॉलिक्युल्स. तर त्याच्यामध्ये डिस्क्रिमिनेशन बिटवीन सेल्फ अँड नॉन सेल्फ. And last characteristic or feature, that is the memory. When the immune system encounters a specific foreign antigen for the first time, it generates an immune response and eliminates the invaders. This is called first encounter. The immune system retains the memory of first encounter, and as a result, second encounter with same pathogen brings about quicker and stronger immune response. म्हणजे अशा पद्धतीनं हे युनिक फीचर्स आपण इम्युनिटीचे स्टडी केलेले आहेत, ॲक्वायर्ड इम्युनिटीचे. ॲक्च्युली आता त्याचे टाईप्स बघायचे आहेत आपल्याला. सो द ॲक्वायर्ड इम्युनिटी हॅज टू टाईप्स. Active, first type, active immunity. The resistance developed by individual as a result of antigenic stimulus, that is active immunity. It may be again two types. Natural acquired active immunity, first type. It is acquired due to infection. It is developed after entry of pathogens in the body. It is long-lasting immunity. त्याचा इफेक्ट भरपूर लॉंग लास्टिंग असतो. फॉर एक्झाम्पल, person who has recovered from attack of measles developed natural acquired active immunity to measles for the lifetime. बघा फ्लो चार्टमध्ये ॲक्टिव्ह इम्युनिटीचे दोन टाईप्स आपण स्टडी करणार आहे. एक नॅचरल अँटीबॉडीज डेव्हलप होतात इन रिस्पॉन्स टू इन्फेक्शन. नाव द सेकंड पॉईंट, दॅट इज द आर्टिफिशियल acquired active immunity. In this, antibodies developed in response to vaccination. म्हणजे actually it is resistance induced by vaccine. Vaccine introduced in body formation of antibodies. आणि ह्याचा वेल नोन एक्झाम्पल आहे, दॅट इज पोलिओ व्हॅक्सिन, BCG व्हॅक्सिन. आणि ही इम्युनिटी जी आहे इट मे बी टेम्पररी or permanent. Then second type, that is the passive immunity. It is acquired when ready-made antibodies are received by the body cells. That means the body cells do not take any active part in the production of immunity. Passive immunity is again of two types. First one is natural acquired passive immunity. Before birth, maternal antibodies transferred from mother to fetus through placenta. रिप्रोडक्शनमध्ये आपण स्टडी केले की मदर आणि फिटस ह्याच्यामधलं कनेक्शन जे आहे दॅट इज थ्रू द प्लासेंटा. आफ्टर बर्थ antibodies are transferred from mother to infant through colostrum. Colostrum is nothing but the first milk of mother. Through the colostrum and continue throughout the period of breastfeeding. But the antibodies received by baby from mother remain in the body for a short time. Therefore, natural acquired passive immunity is short lived. So, see in the diagram again, passive immunity which develops after antibodies from someone to somewhere else. It is divided into two types: natural, that is, antibodies received from mother through the breast milk. Now we study the second type, that is the artificial acquired passive immunity, in which antibodies received from a medicine, for example, from a gamma globulin injection or infusion. with example आपण हा टाईप आता स्टडी करणार आहे. It is developed, that means the artificially acquired passive immunity developed by injecting previously prepared antibodies using serum from humans or animals. For example, antibodies obtained from hyperimmunized horses. That antibodies are injected to humans against rabies pathogens. It is also short lived. Then, second type of the immune system, that is the cells of immune system. So, in the type of cells, there are actually two important cells which are concerned with the immune system. First one is the lymphocyte and second one is antigen presenting cells. First we study the lymphocytes. Lymphocytes are the main cells of immune system. They arises from bone marrow, undergoes differentiation in gut, which is associated with lymphoid tissues, that is tonsils and Peyer's patches. They are also called as the bursal cells or B-lymphocytes. And other cell is called T-lymphocytes, which are formed in the thymus gland. The B-lymphocytes forms humoral or antibody mediated immune system, commonly known as antibody mediated immune system AMIS, and T lymphocytes form cell mediated immune system, that is CMIS. Thus, related with these two cells, B lymphocyte and T lymphocyte, we study the mechanism of response of T lymphocyte to antigen. So, coming in contact with antigen, T lymphocytes form clones, same copies, of T cells which are similar but perform different functions. The clones has four types of T lymphocytes. First is helper T cell. Sensitized helper T cells produce lymphokines for perform different functions like proliferation, stimulation of B lymphocytes and macrophages. Second type is killer T cells. These are also known as cytotoxic cells. They are directly attack and destroy invading microbes, infected body cells, and cancer cells. They also bind to infected cells and secrete perforins. Then perforins form a hole in infected cell and releases substance that kill the cell, hence the name cytotoxic or killer T cell. third type is suppressor T cell. These cells suppresses entire immune system against attack on the own body cells. And last type is memory T cells. These are previously sensitized cells which retain the sensitization memory for long time in the future. Then the second type of mechanism of action of B lymphocytes to antigens. B lymphocytes are sensitized directly by antigens and helper T cells. Activated B lymphocytes multiplies very fast to produce clones of plasma cells and memory B cells. Plasma cells produce antibodies and circulated in blood and lymph and bind to cell membrane or remain free. Free antibodies have three main functions. First function is agglutination of particulate matter like bacteria and viruses and engulfed by phagocytosis. Second function, opsonization or coating of bacteria to complete phagocytosis by macrophages. And third, neutralization of toxins released by bacteria. For example, टिटॅनस टॉक्सिन म्हणजे धनुर्वाताचं इन्फेक्शन होतं त्याच्यामुळे टॉक्सिन फॉर्म होतं ते न्यूट्रलाइज करण्याचं काम बी लिम्फोसाइट्स करतात. Then the next point in the cell immune system, that is the antigen presenting cells. Actually, antigen presenting cells engulf invading pathogens and process the antigen. So see in the diagram, the processed antigens are present on their own surface. सेल्सच्या सरफेसवरतीच ते अँटीजेन्स प्रोसेसिंग चालू आहे. These cells are able to deliver a stimulatory signals that is necessary for activation of helper T cell. That's all about the immune system and the cells which are responsible for protection of our body from the foreign particles or pathogens in the form of microorganisms. Thank you.